Hello everybody and welcome back. So, we are discussing about the carbohydrates and carbohydrate based molecules that can have various applications. And on that course of discussions, we have come to the synthesis of or development of nanomaterials or nanoparticles that I have talked about in the last lecture, that the macromolecular carbohydrates that contain a long polymeric chains, sometimes the modifications. This one was our chitosan, which has amine groups as branched chains in its polymeric chain structure. And in the last lecture, I have talked about that how you can use these kinds of functional groups to do various chemical modifications for your biological applications. And I have also talked about a little bit that you can synthesize the nanoparticles or the nanomaterials out of these molecules by themselves. So, basically I will start today with the importance of the nanomaterials, or now which we can call nanomedicine, or nanobiotechnology, you can say. Even nanochemistry. So, that basically covers the area of using or developing new nanomaterials or new nanoparticles or nanomaterials for biological applications, such as drug delivery, such as using the molecules themselves as the medicines, such as in order to study several binding property activities inside the cells, taking probe molecules into the cells in a nanoparticle format. And since we are talking about this, the most abundant amount of nanomaterials or nanoparticles that has gained enormous importance are the metal based nanoparticles. So, I will just talk very briefly without these metal based nanoparticles. I think nanobiology or nanobiotechnology or nanomedicine is incomplete. So, I will very briefly talk about this and then we will conclude. Metal based nanoparticles have gained a lot of importance in nanochemistry in general and particularly nanomedicine. For example, silver nanoparticles, silver nanoparticles show very good amount of antibacterial property. Similarly, gold nanoparticles they have actually For certain types of cancers, they show pretty good amount of anti-cancer activity. So, they are used as therapeutics. So, they have therapeutic activity to kill certain cancer or tumor cells. So, gold nanoparticles, in literature you will find there are many different types of gold nanoparticles that have been synthesized, that have been developed, and people are doing research on them. Many of those varieties show pretty good activity against the tumor cell lines. Similarly, nowadays copper mediated nanoparticles, especially copper based nanoparticles, especially the copper nanoclusters, copper nano clusters. They show very good biological activity, antibacterial, anti-tumor both. In general, I will write biological activities. Lot of cellular studies have been performed using the copper nanoclusters. And today a lot of people are trying to develop even iron nanoparticles. Iron nanoparticles have one particular property, that they are magnetic in nature. So, they are usually being called magnetic nanoparticle. Magnetic nanoparticles, and they are being used to treat certain types of virus, to kill virus and bacteria. Because of this, their magnetic properties, they are being used to destroy certain types of viruses that have themselves certain magnetic activity, or the bacteria. So, these are some of the examples of the nanoparticles being used in medicinal chemistry or nanobiotechnology. So, now the question is how these nanoparticles are usually synthesized. So, as I have mentioned, nanoparticles usually have the size range one nanometer to, you can say, up to 1000 nanometer, but for good biological activity, it usually should be between 1 to 100 nanometer particle size. Nanoclusters have larger size than 100 nanometer, but they fall within 1000 nanometer, maybe 300, 400 nanometer sometimes. So, what you are actually doing here is you are using certain components, you are using a chemical, for example, this metal things, metal based compounds, and you are reducing the size of the metal. That is done usually. So, for example, if you take silver, if you consider synthesis of silver nanoparticles, then you take a salt of silver, maybe a silver nitrate salt. So, this is a salt where silver exists in +1 oxidation state. Now, if you can reduce this under controlled manner, reduction, then you form silver 0. And if you have a particular type of environment here, environment there, that actually forces these particles to reduce their size or to aggregate or disperse, but it will reduce their size, and that is when it forms a nanoparticles. So, silver 0 oxidation state will exist as silver nanoparticles, Ag nanoparticles, which will be surrounded by its environment, and most often the environment is the reducing agent that you have used. That is the first environment, actually surrounding environment. So, you can use chemicals. Typically, if you want to reduce silver nitrate to silver 0, you can use reducing agents such as sodium borohydride, then it will be surrounded by counter anions, and that will be the first reason for the formation of silver 0 into nanoparticle form. Or quite often, once these nanoparticles are forming, they are not quite stable. So, you need a certain stabilizing agent. And here we usually use large macromolecules. So, that will further stabilize the nanoparticles. I will show you one example. So, this is your stabilized nanoparticles. For example, if you want to synthesize gold nanoparticles, typically people start with gold aurichloride, HAuCl4. It's a liquid. Make a solution of gold aurichloride, and then you use a reducing agent here, sodium borohydride. So, you take a test tube, add gold aurichloride, add sodium borohydride in a common medium, keep on stirring for some time. It depends upon your reaction conditions, it can take from minutes to hours, but it finishes usually typically either half an hour to 1 hour, it will be done most of the times. Then you will have gold 0, and then if you use certain stabilizing agent, the most common stabilizing agent people use is citrate. Citrate is basically citric acid structure. This is OH, COO minus, COO minus, I think COO minus. So, that way you can surround the gold with the charge density, and here you will have all the negative charge on the surface because of the carboxylate anions, and that is the reason why gold nanoparticles are formed, and gold nanoparticles have usually very small size. You can even purchase nowadays gold nanoparticles from 5 nanometer up to 20 nanometer or maybe 50 nanometer. So, it can be as small as 5 nanometer, it can go large also. So, I will show you, this is how it looks like, but this is not citrate stabilized, this is the gold nanoparticles we have synthesized in our laboratory. This is basically we have done using the polymer like chitosan we have used. So, this is how it looks like when the nanoparticles are synthesized. This pink color actually demonstrates that your nanoparticles have been formed. And in this case, the nanoparticles are dispersed, well dispersed in the solution. It is not precipitated out, because this is typical for gold, because gold nanoparticles are so small that you cannot usually precipitate it out. They are dispersed in solutions and they show color. The reason for this color is you can read a little bit, that is surface plasmon resonance, because of particle particle interactions. Basically, surface plasmon resonance is the reason why the nanoparticles show colors, and you can pretty much see them in, if you take a UV visible spectroscopy, you can see that before gold aurichloride does not show any UV band. Once you started synthesizing the nanoparticles, a new nice UV band will come up, that is the responsible for this color, and this band is because of the surface plasmon resonance band. So, the problem here is, if you want to use these nanoparticles for biological purpose, then these nanoparticles have to be non-toxic. That is the first criteria. And when, as I have said, when you are using a reducing agent, the reducing agent stays on the nanoparticles. It provides an environment, it provides an interaction factor. So, it stays with the nanoparticle that is synthesized, and these reducing agents are toxic. These are pure chemicals. So, ideally the nanoparticles that are being developed using the chemicals as the reducing agent are not good for application into biology. So, nowadays that is why people use natural products such as chitosan, such as aloe vera, such as silk, such as citrate. Citric acid is also a natural product obtained and does not show much toxicity. So, use of those materials as reducing agent, and they also act as stabilizing agent also. So, I will show you, just for example, if you take the example of chitosan reducing agent plus stabilizing agent both. So, if this is your chitosan, you have amine group here, amine group there, and here you have the hydroxyl groups as well. So, those hydroxyl groups are also responsible for reducing your nanoparticles. If you use gold aurichloride and without the reducing agent, you can develop a method without using other chemical as reducing agents. It will, you have to do certain kinds of optimization to use this. Then you will have gold nanoparticles surrounded by chitosan. Here would be amine, here would be amine. So, positive, basically when you are using this acid, so gold aurichloride is basically have H plus also, right. So, that will protonate amine into NH3 plus, and those cations are responsible for stabilizing the nanoparticles, that forces the particles to be in the nanoparticular form. And on the surface you can have the other ones, and the unreacted amines can be there, hydroxyls that are present, they can be there. So, depending upon what agent, what polymeric compound you are using, it can be chitosan, it can be PEG, which is polyethylene glycol, there are many, it can be peptides. You can synthesize nanoparticles with a stabilizing agent, and they are supposed to be non-toxic in nature, because whatever you have used throughout is kind of natural units. And such kind of nanoparticles have advantages. Number one is, of course, I have talked about the nanoparticle itself for example gold has anti-tumor activity. So, you can use this itself to kill the tumor cells. Number 2, since you have stabilized or since you have covered your nanoparticles or nanoparticle are merged with the macromolecular structures, you can do lot of chemistries on the surface of the nanoparticles. So, in here you have hydroxyl, if you use something else you can have carboxylic acid end here, you have already amine ends here. So, you can attach the targeting moieties or other kinds of components that you want. You can attach a peptide here, you can change its viscosity, you can change its solubility property by adding either the hydrophobic group if you want it to be more insoluble. You can add more hydrophilic group in order to make it more dispersed or more soluble. You can add DNA to it. Lot of studies have been done, lot of nanoparticles have been synthesized where DNA are attached to study different activities including the single point mutations. Gold nanoparticles have been enormously used and very nicely used actually to find out presence of single base mutation in DNA. And gold has another kind of property that when they are in dispersed mode, gold 0 nanoparticles, not silver, gold. So, gold nanoparticles in disperse, they show pink color as you have seen this color. So, that is the dispersed nanoparticles, pretty transparent you can see. Now, if you vary the concentration and make it more dense, this is also gold nano, but aggregation, because of high concentrations the atoms aggregate, the nanoparticles aggregate. So, this is aggregation. The reason for aggregation are various. It can be atom-atom interactions, or it can be the interaction between the surface. The functional groups present in the surface can interact between each other also. So, these are the functional groups present in the surface. So, depending upon your choice, you can make your surface in such a way that it will facilitate aggregation. So, for example, those who are biologists may be knowing that biotin avidin binding is very strong, and people in biotechnology, in chemical biology, this is one of the oldest technique. If you want to study binding between two components, one component is attached with a biotin, other component is attached with a avidin, so that you know that they will come instantly closer together. So, here also such kinds of tricks have been used to make the aggregation. So, if you have a mixture of biotin stabilized gold and another one is avidin stabilized gold, then if you mix them together they will immediately form aggregation. So, you will have gold aggregation, and this aggregated gold has a color of intense blue. Intense blue color you will get. So, this change of color you can measure of course using the spectroscopic techniques, using the UV. This change of color is also a way to study different biological aspects, and this is what has been used very nicely to find out presence of single base mismatch in DNA. For example, if one gold nanoparticle you attach with a single stranded DNA and another gold nanoparticle you attach with another ssDNA, single stranded DNA. If these two are complementary, then you will have hybridization, perfect hybridization, and that will give you aggregation. So, it will show you blue color. This is pink, this is pink, gold mixed together, that will give you blue color. If these are not matched properly, If there is presence of a mutation here, then they will not aggregate. So, and you will not see the blue color. So, like that, this is the blunt way I have explained, but there are a lot of tricks in it. So, these kinds of phenomena have been used very nicely to study lot of biological activities, lot of chemical activities as well. So, like that you can develop many nanoparticle materials. And one important thing I forgot to mention, I should mention, is that you have stabilized this with the polymeric material. Another reason of using such bulky large structured material is that because of their hydrophilic property, or sometimes they have hydrophobic property, you can make use of them to encapsulate or to trap small molecules such as drugs. I have talked about using only the chitosan or only the macromolecule and the drug. Now, I am talking about using the metal nanoparticles stabilized by these macromolecules and encapsulated drugs. This is where the most of the drug delivery applications came. So, you can have if you have your drug hydrophilic, then it can easily be entrapped on the surface, because these are hydrophilic in nature now to some extent. So, your drugs can be encapsulated here, and then whole thing as a nanoparticle form will get into the cell. If your drug is hydrophobic, then you can make these things hydrophobic by attaching the long carbon chain, hydrophobic carbon chain, and then your drugs would be inside, will go inside. If these are hydrophobic in nature, drugs can be bound here, So, and the whole thing stays as a nanoparticle kind of form, quite stable, and then you can take that into the cell. So, drug delivery applications are huge when it comes to metal based nanoparticles. All right. So, with this I will conclude this module. This is how the size of the nanoparticles look like. This is the 200 scale, this is called FETM, Fourier electron transmitted electron microscopy. Transmitted electron microscopy is TEM, and FETM is the Fourier transform factor. So, this is the size of the nanoparticles, this is the size of the nanoparticles, This is some of our picture actually. If you look at a single nanoparticle, this is the size, which means this basically is around 50 nanometer. So, this is well within 100 nanometer range, and we have synthesized by modifications of the chitosans with the drug there. So, like that if you read it in a high end machine, it will show you the image like this. Okay, so with this I will conclude this module, and today I guess is going to be my last lecture of this course. So, I will give you a recap of whatever we have covered in this course so far. So, the title of this course obviously is Essentials of Biomolecules, Nucleic Acids, Peptides, and we have later on added the carbohydrates. I have divided into 10 modules, the last one being the module 11, which is the recap basically. And over time, the first course content that I had shown you, we have done kind of nominal modifications in 1 or 2 modules, changed the names for example. So, this is the total overall module features that we have covered. First one being nucleic acids and proteins. Second module was on synthesis of artificial nucleobases or as well as natural nucleobases and nucleotides. Third module is DNA replication, polymerases, DNA sequencing, and PCR. Module 4 was on DNA damage, mutations, and cancer. Module 5th, we have talked about DNA to proteins, the process of transcription, translation, and the genetic code. Module 6 is about protein sequencing methods. So, I had divided the module into two. It was a single module. These two, 6 and 7 was single module. So, module 6 is on protein sequencing methods. Module 7 we have talked about peptide synthesis, the organic synthesis of peptides and its application into therapeutics. Module 8 we have talked about certain modern techniques that we use in chemical biology for diagnosis of disease. Module 9 was on molecular probes, PNA and LNA. Module 10 was the this module that we have just finished is the chemistry of sugars and carbohydrates, and how we can use the carbohydrates as biomolecular probes. So, I will give you a brief overview of whatever we have learned so far, the key points and the key aspects of those modules. So, let us start with module 1, nucleic acids and proteins. So, in this module we have actually seen how the double helix structure of DNA was discovered or evolved, taking all the informations, taking all the right kind of informations that were available that time. For example, it was started with the Miescher, who has first isolated the DNA contents, the units phosphate, sugar and base, and then Charga found out that the equivalency of A and T and the equivalency of G and C, number of adenine would be equal to number of thymine, that number of guanine was equal to number of cytosine, and this equal factor exists in all DNAs that he has isolated. So, and using these two key informations and using the book that gives the bond distances, that gives the angle, that talks about the electronegativity of the atoms, Watson and Crick has finally established the double helix structure of DNA, taking the experimental crystal X-ray diffraction data that was given by Rosalind Franklin and Maurice Wilkins. And so, this is the double helix structure basically, which is actually an right handed helix. The key parameters or features of a double helix DNA, or we call it B form of the DNA, is that the distance or the diameter of the helix is 20 angstrom. The distance between two nucleobases, so nucleobases are perpendicular to each other. So, here is one nucleobase, here is other nucleobase, as it is given here. This is the distance between the neighboring nucleobases. So, they are in this form, is around 3.4 angstrom, and in a helix pitch that distance is 34 angstrom. That means one complete turn is 34 angstrom, which means each helix pitch or each complete turn contains about 10.5 base pairs on average. And because of the double helix structure or helical pattern, it forms two kinds of grooves. One is called the major groove, which is explained here, that is a large cavity, and the major groove, because it is large, it is for, or it allows binding with large other biomolecules such as proteins, such as other nucleic acids like RNA. It also has a minor groove with a small cavity here, and that is being small, it only allows binding with the very small molecules like the ions, like the drugs, like other small organic molecules. So, these are the key features or the key parameters of the double helix. This is where a DNA stays inside the chromosome that we have talked about, stays inside the nucleus. When it comes to the structures, this is the how the structural orientation happens. This angle, it has a dihedral angle minus 60 degree. So, that is very important for a DNA, that makes the DNA helix actually. That is the backbone structure. Sugar is oriented little bit tilted from the backbone, or sometimes you can just think of like this. If this is the backbone, this is your sugar, this is your sugar, and then the bases is like this, nucleobase is like this. When it comes to the conformation of the sugar nucleobase, syn geometry, where this the nucleobase will fall on top of the sugar is not allowed as it is. This is the actual geometry, anti-geometry that exists in DNA. Similarly, the sugar pucker conformation exists as C2 prime endo twist for DNA and C3 prime endo twist in RNA. And because of this, it can attain the AT base pairs and GC base pairs with 2 hydrogen bonds and 3 hydrogen bonds respectively. Then after the DNA, we have talked about the amino acids. We have seen all the natural occurring amino acids, their structures, their variations, as we have talked many times I think. And we have seen how once you have the sequence of a protein or sequence of a peptide, you call it the primary structure, then it does not stay in that sequence forever because of its single bond rotation. Here is also a single bond. Because of the presence of the single bonds, the bonds can rotate and it can assume certain orientations. That is called first is of course we call them the secondary structure, that is because of the local interactions. And we have seen that it can assume 2 kinds of conformations. One is the helix structure, which is like this. This is a picture of the collagen. It can either be an helix or it can assume beta sheet conformation. So, that is a planar conformation. So, this plane can be of 2 types, one is the parallel, another is anti-parallel as it is mentioned. And the beta conformations are basically because of the intermolecular hydrogen bondings and pi stacking. Alpha helix was because of the intramolecular hydrogen bonding interactions and electrostatic interactions. So, these are the secondary structure of the amino acids or secondary structure of the proteins. And then large scale interactions, little bit distant interactions gives a folded structure or a 3-dimensional structure of the protein. That is because of the variation of the side chains that are present in the protein. So, hydrophilic side chains are there, hydrophobic side chains are there, basic side chains, acidic side chains, all these factors are there, and because of their long range interactions they kind of fold into each other. That gives you the 3-dimensional structure or the tertiary structure of the protein. And as a result it forms hydrophobic core, it forms the hydrophilic surface. Usually we have seen the interior of the protein is hydrophobic in nature, that does not allow water to come in. The surface of the protein contains all the polar side chains or the charged side chains, and that interact with the polar solvents such as water or other electrostatic molecules. And we have seen also that from unfolded secondary structure of the protein to the folded tertiary structure of the protein, this is a very spontaneous process. So, this is the ultimate geometry or ultimate conformation that a protein exists. So, those were about module 1. Now, coming to module 2, second module, we have seen how we can develop how the nucleobases that are present in nature or you can make lot of different modifications for our own purpose. And how can we also synthesize the real nucleotides such as DNA in your laboratory. So, these are some of the methodologies that we have talked about. This is the synthesis of pyrimidine compounds in general, that basically means the synthesis of the pyrimidine nucleobases also, such as thymine, uracil and cytosine. This is how you can synthesize the purines, adenine and guanine and their various derivatives. These are the techniques how you can synthesize the nucleosides, which means attaching the bases onto the sugar. That has various 2 or 3 approaches we have talked about. This is one of the example how thymine, this is uracil. So, uracil was attached with the sugar moiety in a multi-step reactions. And then we went on with the synthesis of the DNA oligonucleotides that we call polymeric structure of the nucleosides or polymeric structure of the nucleotides. So, this is mostly you have talked about the solid phase phosphoramidite chemistry. Phosphoramidite was the monomer this that you have synthesized in using the organic synthesis methods. So, which basically has a sugar moiety attached with your desired nucleobase, and in the 3' end you have a phosphorus with a protected way, and your 5' end also has a DMT protection. So, it is basically about the reaction that will allow you to continue this way to make bonds and go further. So, it goes on a series of reaction cycles, series of reaction steps also, and everything we have talked about, that everything can be done in a machine called the DNA synthesizer. Or you can call it also oligosynthesizer. And this is the place where the synthesis actually happens. It goes through lot of steps, through activation, then coupling, then capping to make sure that the unreacted ones that are remained in the solid phase does not react in the next step, then oxidation, then the cycle continues. And using this technique you can synthesize short oligonucleotides, short DNA sequences, and that can have a sequence up to maybe 30, 40, 50. 50 is little bit too large, but at least 30 base pairs long DNA. And then came a very large module, that was module 3. This is where we have talked about the a biological process that goes on inside the cells, that is DNA replication, and then how we can how we have developed technologies to use the kind of the process of replication to synthesize more copies of DNA. So, it is basically from the cell to a machine. We have also talked about DNA sequencing techniques, and all of these actually use the same kind of this idea of DNA replication. So, in DNA replication is basically how you can multiply, how the multiplication of DNA or gene happens inside our cells. From one DNA, how you can get 2 copies of double stranded DNA. That is what happens during cell division process. One cell divides into 2 cells and each cell has a nucleus. So, each cell contains all the genetic molecules, that is DNA. So, one DNA from the parent cell has to be divided into 2 DNA which is actually the same as the mother one. So, that is what is the process of replication, and we have seen that it goes through a series of steps involving lot of enzymes, involving lot of other small molecules, involving oligonucleotides such as RNA. So, helicase is the key enzyme present, SSB protein we have talked about. The most important enzyme we have talked about was DNA polymerase, and that you need 2 kinds of polymerase for DNA replication. one for the 5 prime to 3 prime strand, other is for the leading strand, other for the lagging strand, that is the 3 prime to 5 prime strand. So, two types of DNA polymerase are essential for cellular replication of DNA, and then other enzymes, of course, RNA primase, RNase, or DNA ligase, and all sorts. And then how to use the cellular process into your laboratory. So, if you need to copy, if you need to make more number of DNA from a given isolated DNA, how can you do that in your laboratory? And that was called the polymerase chain reaction. PCR is the technique where you can amplify the genomic DNA that you have isolated from certain organisms, and we have developed certain steps to make this process possible. You need a DNA template, which is basically the DNA that you have isolated, and then you need the individual components that we call DNTPs, triphosphates, nucleotide deoxyribose nucleotide triphosphates, that are basically the building blocks for the synthesis of the counter strand. And then the key importance was the use of the polymerase, which you have used too for the cellular process. Cells use two, but we cannot. In this case, we have seen that we do not need to use two polymerases. You can use single polymerase, but the problem was that in the process you need to break your double stranded DNA into single strand, and that requires high heat. So, the key question was, will the polymerase be stable in that high temperature? So, you cannot do your reactions at high temperature, because polymerases, the normal polymerases, they are used to function at the physiological temperature, which is 37 degrees Celsius in our body. And there came the major breakthrough that people have found out, the discovery or the isolation of Taq DNA polymerase, that is basically a thermophilic bacteria which functions at a very high temperature, close to 100 degrees Celsius. And this is the Thermus aquaticus, is the organism, microorganism or the bacteria. From that bacteria, the Taq polymerase was isolated, and the name also came from there, the Taq, because of the Thermus aqueous. T AQ is aqueous, that makes it Taq. So, use of Taq polymerase solved your problem of high temperature during the course of the reaction, and then things become easier. Of course, we have learnt already how to synthesize oligonucleotide. You have used that as primers here, and then you could do the cycle of process that will every time multiply your DNA, and that can be done in this machine called the PCR machine. And then one of the best, one of my very favorite topic was the sequencing of DNA. How can you find out the actual sequence of the DNA? And the most I have talked about was the Sanger's dideoxy method, which is one of the best method actually for sequencing a DNA. And here we have seen how a synthesized molecule, which is the dideoxy, was used, dideoxy NTP was used as a stopper during the replication of the counter strand. So, every time in each of these vials you have added certain amount of the DDNTP, and that was actually stopping at every base when you have been elongating the synthesis of the counter strand using the enzyme polymerase. And by studying those fragmented patterns, you can see the in the gel electrophoresis diagram, very nice, every nucleobase will give you a band, break of every nucleobase or the stop of every nucleobase will give you the band, and it is one of the simplest analysis technique that you can imagine, through which you can actually write down the sequence of the whole gene or the whole long DNA very easily. Then module 4, we have seen here the chemistry that actually goes on inside our cells, or that are the main key reasons for the development of mutations, which in turn are the reasons for aging and cancers. So, how the DNA gets damaged, how those damages are responsible for mutations, and how those mutations are adapted to cancer. And here we have of course talked about first with the UV induced DNA lesions, that we have seen that if you have a dinucleotide that can form the cyclobutane pyrimidine dimers by doing a photochemistry, it can be two different types. One is 2+2 cycloaddition reaction standard, other is also a 2+2 cycloaddition, but a different one, Paterno-Bucchi, that gives you formation of the damages like this. And these damages, because once they are the chemical nature of the individual nucleobases are now changed. So, if you have started with T, this is all T, this is no longer 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 T. Here you have a merged one. So, nucleobases has changed their chemical structures, which obviously means that they have changed their chemical properties also. And those change of chemical properties are the reasons. This is for cytosine we have seen that it undergoes deamination also, that gives you conversion from cytosine to uracil is the deamination, and those are the structural changes that are responsible for developing mutations, and those mutations are the key reasons for the growth of the tumor cells or the cancers. Module 5. In module 5, we have seen how the proteins are biosynthesized in cells, and they take the informations that are present in DNA, that are present in the gene. Those informations are used to synthesize or as directions to synthesize the proteins of a particular sequence, and that has two key steps, that is, one is transcription, other one is translation. The process of transcription was how you can synthesize or how you can take the information from the nucleus to the cytoplasm, because protein is synthesized in the cytoplasm, DNA stays in nucleus. So, how this information, transfer of information happens from the nucleus to the cytoplasm, that is basically your transcription. And here we have seen how mRNA was synthesized, taking the genetic code as the template. So, that is the first thing. Second is how mRNA with its sequence, with this information, is governing the synthesis of the protein, or in other words, how the language present in the nucleic acid language, the codons present in mRNA is translated into an amino acid language, which is protein. So, here was the process of transcription that we have talked about, that in the genome you have double stranded DNA, 5 prime to 3 prime, 3 prime to 5 prime, which we have called sense strand and anti-sense strand. So, idea is that you have to this sense strand contains all the information. So, you have to synthesize a copy of this strand. So, best way is that you use the antisense, the cell you had used the antisense strand as the template, and synthesize the complementary mRNA sequence from DNA to RNA, and this mRNA sequence contains the exact sequential copy of the parent DNA, and this comes out of the nucleus. So, that is what was the process of transcription, or this M means the messenger. So, RNA is behaving as a messenger of the gene. Next came the translation, how is the nucleic acid sequence in mRNA is translated into amino acids, and there we have seen the activity of, this is where the amino acid is attached, activity of tRNA, transfer RNA, which has of course a complicated structure and which carries amino acid here, and each of the tRNA carries a 3-letter code, RNA code here, and this is the complementary to the mRNA sequence, part of the mRNA sequence. And we have seen that the whole thing happens in a reservoir which is called ribosome, acts as a platform where all the synthesis happens, and the details of the process we have talked about, that a ribosome has 3 sites, E site, P site and A site, where the mRNA comes and binds. So, each tRNA comes and binds to its opposite mRNA strand, and each tRNA contains amino acid. This tRNA contains also an amino acid. They come close in proximity and then the reaction happens. This attacks, this maybe the peptide coupling happens there. This is where the peptide coupling happens, P site, A site, this, and it goes on, goes on, goes on. And here comes the codon we have talked about, because this is the place the tRNA, this specific 3-letter alphabet binds with the mRNA. So, this is very important, which we called anticodon, and this is responsible for which amino acid will be come in here, and then that we call it a codon. So, codon came into a picture here, for which Professor Corona is very famous. So, here we had given a table chart of which codon is responsible or which codon determines which amino acid sequence. You can have all these codons. This also have a start codon. Without that start codon, the protein synthesis does not start. This is the start codon, and you also have a stop codon. You have to stop somewhere. Once it reaches the stop codon, the synthesis stops there, because the stop codons does not carry the amino acids. Module 6 was on protein sequencing, and here we have discussed how at least two different methods we have talked about. One is Sanger's method again, and another one was what was it? Edmonds degradation method, and third method is of course using the mass spectrometry. So, Sanger's protein sequencing method, Edmonds degradation method and mass spectrometry method. All of these methods are associated with lot of other side reagents to because you have to hydrolyze, we have, that is what we have seen, you have to hydrolyze the peptide sequences, large protein units into the smaller peptide sequences, because otherwise these methods cannot handle such a large length of molecule. So, yeah, Sanger's method was primarily used for N-terminal amino acid determination, and then full sequence determination we have seen that was done by Edmonds degradation method. Module 7 was on again organic synthesis. How can you develop short sequences of peptides in your laboratory, and their applications into therapeutics. Here we have seen that certain peptides show very good therapeutic activities, like RGD peptide, NGR peptide. Then you have, these are basically the tumor homing peptides, so that can get into the tumor cells specifically. And then you have cell penetrating peptides, which are polymeric peptide with a high positive charge density, such as arginine, such as lysine, that they can penetrate into the cells. So, similarly, if you are very important is the blood brain barrier peptides, because it is actually very difficult to take any medicine into the brain cells, because brain cells are overprotective. So, certain kinds of peptides such as this sequence, they are actually, they can invade or they can cross the blood brain barrier. So, such kinds of peptides can be used as therapeutics to carry other drugs along with them into the brain cells particularly. Then we have talked about how the solution phase chemistry you can adapt to synthesize small peptides, but the best one is of course by using the solid phase peptide synthesis protocol, which goes in a multi-step organic chemistry reactions all happens in the solid phase using a resin, and you can synthesize fairly a good length of the peptide from there. Some of the peptides that has been used as drugs that are currently in market, this little bit complicated geometry. This is a cyclic peptide, this is open chain peptide, and there are many peptides still in clinical trials. There are some peptides already in the market, and lot of peptides that we currently do research on for various aspects, to study various aspects. Every day new sequence of peptides are synthesized and their activities have been proven, but they are not yet in the market, of course. Module 8, we have given some descriptions of some of the technologies, molecular technologies as well as instrumental techniques that we nowadays adapt to for biomolecular research into chemical biology, into biotechnology, into bioorganic chemistry, whatever you call it. For example, I have given an overview of the spectroscopic techniques, how UV visible fluorescence, FTIR, NMR I have not talked about. These are the some of the spectroscopic techniques that can be used on the biomolecules. There can be mass spectrometry, we have talked about that, electrospray ionization. We have talked about high end mass spectroscopy, that is mass, MS. We have talked about for very large polymeric biomolecules, macromolecules that has very high molecular weight. You can use the multi matrix assisted laser desorption ionization technique to find their purity or to do their characterizations actually. You can also do protein sequencing using this mass and as well as MALDI technique. And then we have talked about the separation techniques for the biomolecules. Since they are very large molecules, it is hard to separate them. We have talked about HPLC, we have talked about capillary electrophoresis, affinity capillary electrophoresis, and then size exclusion chromatography also, among few others. Module 9 was on molecular probes, how we have synthesized as organic chemist. Our aim is to develop new molecules that can have utilities in biology. So, peptide nucleic acids PNA and locked nucleic acids LNA are the modified versions of the oligonucleotides. They are different than DNA and they can be used to study the biological activities or biological interactions, as well as to find out the presence of mutations in DNA. So, we have described the structure of DNA, how the PNA and how the LNA are different, what are their structural features, what are their advantages, what are their disadvantages. For example, both PNA and LNA, especially the PNA, because it has a peptide backbone, it can invade into the cells very easily. It is not destroyed by the metabolic systems, mostly the nucleus. This is stable, nucleus stable. This is also to some extent nucleus stable. DNA is very unstable to nucleus. Any foreign DNA would be cleaved by the nucleus. So, this, if you want to carry a foreign molecules, these two are the better choices. So, we have seen why that use of PNA, if you use, they make a very strong hybridization with the target. So, that enables it a very good candidate or molecular probe to find out the mutations present in the gene. Then the last module on carbohydrates, where we have seen the structures of the sugars, we have seen the structure of the ribose and deoxyribose. We have talked long back. We have seen the structures of the large carbohydrate macromolecules that are present in nature, such as chitosan, such as starch, such as cellulose, and many others, and some of the drugs that contain carbohydrates also. And we have seen some of the reactions, how you can play around with the chemistry of sugars. And finally, we have talked about how the large macromolecules you can use for medicinal purpose, such as nanoparticles, such as drug delivery. So, this was about the layout and the more or less whole about the course that I have covered. So, and with this I will bid adieu for this course, and I hope that I was able to describe certain methods. I was able to give an overview of the biomolecules, how their structure look like, how their activities occur, how they act inside the cells. And I hope that I was also able to describe certain methodologies, certain developments, the techniques, modern ways, modern techniques that we adopt nowadays in our laboratory to study the biological molecules. So, with this I wish all of you to have good health, and thank you for attending the lecture, and all the best for your future. Thank you.